Итак, мы приступаем. Коллеги, наша сессия посвящена деятельности особо охраняемых природных территорий, которые функционируют в Арктике. В первую очередь, конечно, это наши, чтобы не обидеть других, все-таки наиболее значимые особо охраняемые природные территории федерального значения, а также у нас есть достойные особо охраняемые природные территории регионального значения, особенно в таких значимых для сохранения биоразнообразия и белого медведя регионах, как Чукотка, Якутия, конечно же, Архангельская область. Поэтому сегодня мы рассмотрим, чем же занимаются наши особо охраняемые природные территории, в первую очередь в отношении сохранения белого медведя, а также его мест обитаний. Но у нас остался один спикер с предыдущей сессии, которому мы, пока он на связи, хотели бы представить слово. Это Дайскэ Hirata, представитель, научный сотрудник Центра вычислительной биологии Санкт-Петербургского университета Петра Великого, и он выступит с докладом об изучении генетического анализа белого медведя. Господин Hirata, вы с нами на связи?
Can you hear my voice?
Вы можете слышать, пожалуйста?
Can you hear me?
Вы меня слышите вообще?
Прекрасно, мы вас слышим и представляем вам слово. Очень рады, что вы с нами.
Hello? Can you hear me?
Немножко погромче, если можно.
Будет speak up a little. That would be great if you could speak up a little.
Hello?
Прекрасно. Вы можете стартовать презентацию.
Okay, we can start your presentation.
Hello, can you hear my voice? Hello?
Господин Hirata, вы с нами?
Mr. Hirata, are you with us?
Yes. So, good morning, everyone. First of all, thank you very much for giving me a chance to talk about our project today. I'm Daisuke Hirata from Polytech in St. Petersburg. Today, I'd like to introduce our project. Okay, firstly, I'd like to explain the background of the project. From the late 90s to the early 20th century, human activities in the Russian Arctic dramatically increased for development. Human activities such as past overharvesting and habitat fragmentation pose a serious threat to the genetic diversity of polar bears. The impact of human activities in the Russian Arctic on the genetic diversity of polar bears remains poorly understood. To tackle this question, we applied a museum genomics approach. Museum genomics is a field of research that applies genomics tools developed for ancient DNA analysis to study biological specimens preserved in museum collections. There is an advantage that it is possible to directly analyze historical DNA samples in the past centuries. However, DNA from museum specimens experiences physical and chemical damages. As a result, small amount of DNA is preserved. The extracted DNA is highly degraded and contaminated with bacterial DNA. Although technical difficulties exist, museum specimens are now becoming genetic time capsules. In our project, we first created an information resource detailing the complete polar bear specimen collection at the Zoological Institute of the Russian Academy of Sciences in St. Petersburg. Then, we analyzed historical DNA of the museum specimens to reveal the genetic diversity and population structure of polar bears in the Russian Arctic prior to industrial development. For sampling museum specimens, a handwritten specimen catalog containing metadata about the specimens was digitized, and photographs of the skulls along with their labels were taken. Based on the metadata, teeth from skulls were sampled for genetic analysis. Polar bear collection of the Zoological Institute has been cataloged and made available in an open access database called BearBase. Photos and metadata such as sampling location and the collection time of the specimens, along with genetic variation data, can be checked in this database. These figures show spatial and temporal distribution of 107 samples used for genetic analysis. Samples were collected across the Russian Arctic from Barents Sea, Kara Sea, Laftet Sea, East Siberian Sea, and Chukchi Sea. Samples were collected at each geographical location over a long period of time, from the late 90s to the late 20th century. Most of the samples were collected before the polar bear hunting ban was implemented in the Russian Arctic in 1956. After sampling teeth from skulls, DNA was extracted from each sample. From the extracted DNA, DNA libraries were prepared, and these libraries were pooled, and this pool of DNA libraries was sequenced by high-throughput sequencer. Sequenced reads were computationally processed and aligned to the reference polar bear genomes. This sequence alignment was used for population genetic analysis to reveal genetic diversity and population structure. Our preliminary results about database creation and population genetic analysis were published in papers. As an ongoing analysis, we are now analyzing modern DNA samples collected from field trips and stored at Marine Mammal Research and Expedition Center in Moscow. In conclusion, how does this research translate into real-world conservation impacts? First, empower strategies with genetic insights. The dataset links human-driven pressure and climate shift to genetic changes in polar bears, enabling evidence-based prioritization of at-risk subpopulations. Second, equip conservationists with tools. Identifying genetic erosion patterns enables developing diagnostic panels and field tests to monitor population health in real time. Third, expand beyond polar bears. The framework combining historical DNA with modern genomics is being adapted for walruses and Arctic foxes, supporting multi-species conservation and coexistence of programs. Yes. Lastly, I'd like to thank the project team members and the financial supports to our project. That's all for my presentations, and thank you very much for your attention. That's all.
Спасибо, господин... Да, спасибо, господин Хирата. Очень интересные всегда исследования генетической структуры белого медведя, тем более для того, чтобы понять вообще популяционную структуру белого медведя.